This video covers the general life cycle of mosquitoes and diagnostic characteristics. Mosquitoes are vectors for several diseases including malaria, lymphatic filariasis, dengue, West Nile, yellow fever, and Rift Valley fever. Malaria is a major public health disease in most tropical countries. It is transmitted from one person to the other through the bite of the female Anopheles mosquito. The male Anopheles feeds on nectar and plant juices and does not transmit malaria. There are about 4,000 different types of mosquitoes in nature. However, only 40 known species of Anopheline mosquitoes are vectors of malaria. In Latin America, Anopheles darlingi, Anopheles Nunez tovari, and Anopheles albimanus are the primary malaria vectors. There are other species that may be of local importance in disease transmission in certain areas. Correct identification of the target vectors is critical to the successful implementation of any control strategy. There are 4 stages in the life cycle of a mosquito, beginning with an egg, larva, pupa, and then the adult stage. The adult Anopheles female mates once and continues to lay eggs throughout its 2 to 3 week lifespan. Anopheles eggs are laid singly directly on water in batches of about 50 to 200 and are unique in having floats on either side. Culex eggs are usually distinguished from Anopheles because they are laid together to form a raft. Eggs hatch within 3 to 4 days, although hatching may take up to 1 to 2 weeks in colder climates. Aedes eggs are laid singly in damp soil. A special feature of Aedes aegypti eggs is that they can lay dormant for about a year and then hatch when they come into contact with water. The main parts of mosquito larvae consist of a head, thorax, and abdomen. The larva has a well-developed head with mouth brushes used for feeding on algae, bacteria, and other microorganisms. The Anopheline larva has no siphon or breathing tube and rests parallel to the water surface. They breathe through their specialized short tubes lining their body known as spiracles. In comparison, the Culecine larvae have breathing tubes and hang down from the water surface. There are 4 developmental stages of larvae known as instars. Factors such as the water temperature affect the time required for development. It is shorter in warmer waters. Anopheles mosquitoes prefer particular breeding sites which are covered in the Entomology Field Techniques video, Collection of Larvae and Pupae. The development from larva to pupa lasts about 3 to 4 days in normal tropical temperature. The mosquito pupa is shaped in a comma and stays at the surface of the water. As with larvae, pupae must come to the surface frequently to breathe, which they do through a pair of respiratory trumpets. No feeding goes on during the pupal stage, during which there is a major transformation from living in water to emerging and living out of water. Anopheline pupa are relatively difficult to distinguish from other species in the field, therefore it is necessary to wait until the adult mosquito emerges for identification. The ability of field workers to identify mosquito pupae from larvae and other insects is important. When possible, field workers should try to separate pupae from larvae collections unless otherwise instructed. The pupa stage takes about 2 to 5 days before the adult mosquito emerges. The adult usually emerges from the pupa at dusk and then rests for a short time in order to harden its body. Shortly after emergence, the mosquitoes mate. The males form large swarms, usually around dusk. Females fly into the swarms to mate. Females may take a blood meal every 2 to 3 days. Blood is needed to develop their eggs. A female will lay a batch of eggs before it takes the next blood meal. In general, adult mosquitoes are easy to identify from other insects. However, care should be made when distinguishing between male and female mosquitoes. Male mosquitoes can be distinguished by their long hairs on the antennae, which give it a bushy appearance. Antennae of the female only have a few short hairs. There are 3 main features that can be used to distinguish between Anophelines and Culecines. the maxillary palps, the wings, or the resting position of the adult mosquitoes. Only female species of the genus Anopheles have maxillary palps that are as long as their proboscis. The Culisine proboscis is much longer than its palps. The wings of the Anopheles species have characteristic pale and dark bands at the anterior. Culex or Aedes mosquitoes do not have wings spotted with dark and pale bands. Adult Anopheles mosquitoes tend to rest at an angle of between 50 degrees and 90 degrees to the surface. Culisines tend to rest with their abdomen about parallel to the surface. Duration from egg to adult of Anopheles may vary between 7 days at 31 degrees Celsius and 20 days at 20 degrees Celsius. A number of taxonomic keys are available to assist in mosquito identification. It is also possible to identify particular mosquito species through biomolecular tests, which can be performed in a reference laboratory. You have now seen an overview of the mosquito life cycle and characteristics used for identification. For more information on integrated vector management or training materials, please visit our website.